Hi, today I would like to discuss on a topic called zebrafish. Actually, zebrafish is a word which you normally would have been hearing this word in pharmaceutical research and on biomedical research. But very few people might have seen this zebrafish in reality, or maybe they might have worked on the zebrafish. So very few people might have hands-on experience on this, and this has become a very good model and also become very famous nowadays in this medical research. It's because it's having lots of applications. I would like to just give a brief description on zebrafish and what are some few applications, which are not at all, I'm not talking about whole of ideas of this zebrafish, but I would just talk on very few applications, and there are still lots of applications from zebrafish. But I would like to discuss on some few techniques which few applications which are most widely used in the pharmaceutical companies and in institutions. So this is how zebrafish looks like. The zebrafishes are very small, and they live in the Himalayan regions in the Nepal and Pakistan, Burma, Nepal. It is found in the Ganges River. It is a freshwater fish, and it grows to a length of 4 to 6 centimeters in length. And the females and males are distinguished by the body shape. Actually, the zebrafish looks like they have stripes, blue stripes running across on the body, so that it looks like a zebra. That is the name called zebrafish came from. And the females would have a pouch underneath, unlike zebrafish males. So that is why we can clearly distinguish males and females in zebrafish, and that would be very helpful for us to take them for breeding purpose. And later we want to have some eggs for research purpose. We can distinguish it easily. And along with that, why we are choosing zebrafish in this medical research is, why can't we use other animal models or any other animals, like any other than the rats and mice? I don't deny that we should not be using these rats and mice, because these are very essential for purpose of a practical purpose, and also learning some any activity or anything. If you want to see any kind of disease models, we have to see through we have to do preclinical research on these rats and mice. But zebrafish is also a very good model, and in which we can find out the drug which we want to, if any targeting drug is there, we can find out whether this drug is having any toxicity or any efficacy in just in a matter of just few days. It may take a longer time in rats and mice, but we may be getting some good result in rats and mice. We may have some positive data, but in order to find out very fastly, and we need to filter compounds which are toxic and non-toxic, and let them go to the next stage into the rats and mice. We can choose zebrafish as a model, and we can compare with the rats like that. In zebrafish and rats, the breeding age is around 2 to 3 months. In rats also, the breeding age would be starts from a third month. In the zebrafish, once the zebrafish gets in mature stage and they are put for breeding, one zebrafish can produce 100 to 300 eggs, which are very numerous, and we can test a number of compounds for any toxicity or efficacy or any other kind of data to be obtained. In rats, the average litter size is around 8 to 13. So having this very few size, and we may be having some very less sample size. And in the zebrafish, the time between the clutches was 1 week, and in rats it is 20 days. So the same fish can be used for breeding in just 1 week itself. We can use it rapidly, and if we have some few fishes, we can frequently change the fishes for embryo production. And moreover, zebrafish has become very famous because these embryos are transparent, and these are ex vivo fertilized. Once they come out of the embryos from the female fish, they look transparent, and the development also takes place very rapidly. If you have put on the tank, It develops very fastly within hours, and it looks transparent. We can see all organs until it gets into a development stage up to some few days. We can see the heart, lungs, and heart, not sorry, lungs, heart, kidney, not kidney, heart, eyes, somites, and liver, stomach, intestine. All these different organs can be very clearly seen under the microscope. And if we, I would like to just talk about the genes and why we are choosing zebrafish as a model for disease models. Is that in this picture we can see there are 70% of protein coding human genes are, are also related to the genes found in the zebrafish, and 84% of genes which are known to be having some human diseases have a zebrafish counterpart. In this picture, if you can see clearly, observe this picture, we have taken human, mouse, chicken and zebrafish. This is taken from an article, from a Nature article, and we can see some genes, species specific genes like 2963 genes are species specific in humans, and mouse 2, 3, 4, 5, 9, 6, chicken and zebrafish. And in these four of these species, there are 10,660 genes which are common, and when we look at this human, mouse and zebrafish, there are 2,059 genes in common. In the same way, if you just compare human, mouse and chicken, there are 892 genes. So here we give a slight, we have some slight doubt that there are almost double the genes present in this comparison with chicken and zebrafish. And human, mouse and chicken are amniotes, unlike zebrafish is an invertebrate. So this huge variation, huge gap between this 892 and 2059 may be the reason why chicken might have gone through the evolutionary stage, might have lost genes during the evolution and might have lost from the humans to the, might have lost all the genes. So this is about the genes, and maybe you can see the zebrafish having some more species specific genes like 3034, which are very huge. It's maybe because of whole genome duplication. And this is the life cycle of zebrafish, and that is the normal zebrafish, how it looks like. But when the fertilization starts, that is when the egg and the sperm gets fused, the fertilization happens, and from then, if you count from 0 hour post fertilization, it goes up to this 1 hour, 3 hours, the cleavage starts, and at this time the maternal gene expression takes place, and until this, and blastula stages can be seen, gastrulation, and here the zygotic gene expression takes place. In the developmental biology, firstly maternal gene expression takes place, and until the zygote doesn't develop its own transcription machinery, the maternal gene expression would be doing its role. So there would be a stage where the maternal gene expression would be shifted to zygotic gene expression. That is at the stage when the gastrulation takes place. And here we can see there is a Spearman's organizer, which indicates that there is a dorsal to ventral axis position. The arrangement of the embryo in the dorsal ventral axis can be visualized by the observation of the Spearman's organizer. See, in this whole diagram we can see gastrulation, neurogenesis, and the time when at around 14 to 18 hours the somatogenesis takes place. Somites can be formed. The whole organs can be formed at around by the end of one day. So by the end of 24 hours, we can see that the zebrafish embryo would be having a tail detachment and also all the organs developed. See, but at the end of 90 days, we can see the whole embryo gets developed into as adult fish, which we can see in the diagram there. So this is all the development which takes place in just a few hours. It's having a very rapid development, and we can see the whole organs gets developed in just 36 hours. If you want to test any kind of developmental toxicity or any other kind of assays, it is very easy to do it fastly if you want to get results very immediately. So this is the model we can go over actually. Let's talk about one of the toxicity parameter which we normally use mostly in the zebrafish is embryo toxicity. See, in this diagram we can see how zebrafish embryo looks like at the time of 1 hour, 24 hours and 48 hours. See that the top one is the normal one which having normal embryo, and you can see the cells were clearly present during the mitosis takes place in the beginning of development. We can see the cleavage at around, we can see the mitosis taking place, and in the second diagram we can see the somites, clearly seen somites, the notochord, the swim, and the eyes are clearly seen. At 48 hours also we can see that pigmentation all starts. But if any drug like any toxic drug, if anything like is given to the zebrafish embryo, and we need to follow some OECD guidelines. There are some few OECD guidelines. So if we need test a drug in the zebrafish embryo, we can see the lethal effects, like we can find out the parameters like the coagulation, where the fusion doesn't take place properly, and we can see that as a diagram in which is shown here. And lack of somites is also one of the feature. Somites cannot be clearly seen, and the tail bud doesn't get detached sometimes, or sometimes the otolith doesn't be seen, eyes are not properly developed. So these are all the parameters which we can see and observe under the microscope during the embryo toxicity study. So along with this embryo toxicity, there are other studies in toxicity where we can mostly we can find out the drug candidate whether it may be having some any organ specific toxicity. Like we can take cardiotoxicity, nephrotoxicity, hepatotoxicity. When a drug is given at an embryo level stage, and we can observe whether these organs are clearly being seen under the microscope or not. For example, if hepatotoxicity compounds like amiodarone or ethanol are given to the zebrafish embryo, the most morphological feature we can see is that we may hear that hepatic steatosis or otherwise hepatomegaly can be seen. So such kind of parameters also we can observe in zebrafish. We can see the enlargement of the liver, and for example in the nephrotoxicity, the parameter like glomerular filtration rate is also diminished. It gets damaged, and one of the important pump called sodium potassium ATPase pump transporter is also present in the zebrafish. So in like these, there are so many applications in toxicity, like in cardiotoxicity as well. If we look into this picture, the normal zebrafish looks at around, this is around 72-hour post-fertilized embryo, and if you can see in this one, in the first picture, and one of the interesting things is that zebrafish has a heart, unlike other fishes, and we humans have a heart with 2 auricles and 2 ventricles, but zebrafish is having only 1 auricle and 1 ventricle. They would be having a wall between the auricle and ventricle, and it also has a blood vessels all along the body. If we look under the microscope, we can clearly see how zebrafish blood would flow through the zebrafish embryo. That's a very good experience if you want to see. And here the auricle and ventricle are attached with each other, and they are very tightly closed and they are overlapped. So when the zebrafish heart gets its inactivity, we can see the auricle and ventricle are rhythmically contracted and relaxed. thereby pumping the blood all across the body. See, as the toxicity of the any drug having any cardiotoxicity, it may be showing some effects like pericardial edema. The V and A represents the ventricle and auricle. The ventricle would be having on the top and auricle is at the bottom. See, the attachment between the ventricle and auricle is getting separated as the toxicity is getting increased, and there is also an increase in the edema all around the heart. So that shows that it's having some cardiotoxic activity in the zebrafish. So not only that, we can also find out some uh, valvular defects. When sometimes the blood passes through the ventricle to auricle, and at the same time it passes from auricle, from the blood and it goes to ventricle to the all organs, it may not supply the blood properly, and the blood may come back because of some valvular defect, like in regurgitation takes place. This is also clearly seen when any other kinds of drugs like tamoxifen also is a cardiotoxic drug. Very slow heart rate will be present, and this kind of activity also seen very clearly. And this is one of the interesting thing in like oil redo staining. Oil redo is a stain, it's a diazo dye, and it actually attaches to it stains the neutral triglycerides and lipids, normally used in pathology studies and when paraffin blocks are taken, and these bind specifically to these triglycerides and lipids. And in this zebrafish, in this diagram we can see that the normal control fish on the top and from the left hand side is having no staining, and in this diagram actually the experiment which they did is that the SEC63 mutant zebrafish was taken. Actually, SEC63 is an enzyme which is a gene which is responsible for if any mutation is taking place in this gene, there is a polycystic liver disease in the humans, and at the same time this kind of mutation is being seen in the zebrafish, and thereby we can see the liver toxicity. And when this oil redo stain has been applied to the zebrafish embryo, you can see that there is a huge deposits of this stain at this liver region, showing some darkish red. So that indicates that liver is intoxicated, and mostly maybe the liver is having some more lipid particles. Like in hepatic steatosis is a condition which you can see in hepatotoxic drugs. So in this way we can also go for hepatotoxicity. And zebrafish also has some other applications in cancer research. When we look at the word cancer, there are many kinds of cancers, and these cancers also can be induced in zebrafish. And one of the main factor which we can see in cancer is that angiogenesis takes place along with the production of cells. The cancer is a condition where actually there is uncontrolled proliferation of cells. Along with that, angiogenesis takes place because the cells which are already getting increased, they should need some nutrient supply. So the main factor which is responsible for angiogenesis is vascular endothelial growth factor. So when this VEGF is induced, angiogenesis takes place, like sprouting of small tiny blood vessels coming out of the main blood vessel. Like in this diagram you can see there are some sprouts, branches of these zebrafish blood vessels. These are called subintestinal vessels, and we can see how they are branching out. For example, when any vascular endothelial growth factor receptor EGFR inhibitors are given, like sorafenib, regorafenib, cabozantinib, or these kind of drugs given, there is complete block, there may be some partial blockade of these angiogenesis. Sometimes there would be a complete blockage of this angiogenesis, even at the control or even in the untreated cancer zebrafish also, untreated cancer treated zebrafish also, we can see there is complete loss of the zebrafish blood vessels. So that indicates that this gives a very good model for angiogenesis studies. Just we need to do is that we just do the actual protocol of slide protocol is there, and we can look for blood vessels under the microscope, and we can see this kind of pictures. And in the same way, lectin stain is also one of the stain which actually binds to the blood vessels in the luminal part of the endothelial cells. We can see the control fish having a nice blood supply to all regions wherever the blood vessels are there, and it passes, the blood vessels are very clearly arranged between the somites, and whole region is completely having some good blood supply. But in this another one which is maybe having cancer potential, maybe having this apoptosis, there is a very less supply, and this lectin stain was given, there are no blood vessels seen there. So these two assays are very good assays in observing this blood vessel growth, and if you want to look under the microscope. Actually, this lectin stain should be given to the zebrafish embryo directly into the blood, into the intravenous circulation. And along with that, actually, the zebrafish cancer can be induced by using the normal cancer cell lines, like just how we want to use for rodent models, xenograft models in rats. Just like the same way, we can take the cancer cell lines for maintenance, and we need to do some properly. We want to how many cells we want to inject. We have to take that cancer cells using the micro injector, and we can inject into the zebrafish embryo. So when this is injected to zebrafish embryo, the cancer cells has a property of dividing still, and it may metastasize, or it may localize at that position, and may have some bigger size and mass, or it may form like small spots around the zebrafish. So this all can be seen under the microscope, and this whole zebrafish the cancer studies can be done in the first stages of embryonic life, around in the first 10 days of this embryonic stage, because after this the zebrafish may develop the immune system, and cancer is already present, and the zebrafish can think that zebrafish has an ability to reject this cancer line as the xenograft. So again, some errors may take place. That's why in the first stages of life, when the immune system is not completely developed, the cancer studies can be done easily. So when these anti-cancer agents are given, there is a decrease in the tumor size, or it may have some decrease in the angiogenesis capacity, and so any progression like decrease in metastasis. So we can look for any other protein studies, Western blotting, and all kinds of studies we can see during this cancer research. Likewise, there are many applications in zebrafish research, like in the developmental biology. We can study teratogenicity. If any known teratogen is given to the zebrafish, like a thalidomide or a phenobarbital, which actually induce the morphological deformities in the zebrafish, like it is reported in the humans as well. There would be defects in the zebrafish tail, or otherwise the jaw regions, or otherwise the liver may gets degraded, the cardiac region, and somites, notochord, the shape of the body may get deformed, the whole organs may not be completely developed. It depends on at what time of period you are giving the drug to the zebrafish development, at which type of development stage you are giving the drug. So you can use teratogens for teratogenicity studies, and cancer oncology, toxicology, and genetics. You can also knock out genes in the zebrafish, and we can, as I already said, there are many genes which are present in the zebrafish, and they're also present in the humans as well. So, and in the zebrafish, we also know that the zebrafish has a liver, and like in humans, how is we have CYP enzymes, which are mainly helpful in the metabolism of food and drugs. Zebrafish also has this cytochrome P450 enzymes. All the isoforms which are what? CYP1, 2, 3, whatever the isoforms which are present in us, the same isoforms are also present in the zebrafish. So that indicates that zebrafish has the ability to metabolize the drug as well, and also same characteristics of the human how it have. So it's a very nice model, and it's also helpful in neurobiology studies, and we can induce convulsions in zebrafish adult stage, and we can also take out the brain from the zebrafish for any kind of studies, and also in Parkinson's studies. There are many applications in zebrafish in environmental sciences, in regenerative medicine. Yeah, it's a very good model. Actually, zebrafish has the regenerative capacity without using the stem cells. And in evolutionary theory, the immune system, like in zebrafish also has neutrophils, lymphocytes, so white blood cells. So these are also present in zebrafish. That would be useful in studying inflammation studies, and also in infectious diseases like in mycobacteria studies, and many of the drug discovery solutions. Zebrafish gives a very good model for studying this all research, and along with that, zebrafish can be used in diabetes. We can see zebrafish also developing diabetes and hyperglycemia and hypertriglyceridemia. We can take out the blood from the zebrafish and look for these parameters like glucose and triglycerides. When there is an increase in when the diabetes zebrafish is having, it may have some increase in the glucose. Not like us, we having 90 to 120 milligrams per deciliter, but it has a very less. But it has the property of getting the diabetes. We can induce it. So I've just explained all clearly about very briefly on all the areas, but there is still a lot to uh, look underneath this research, and we can find so many articles, and we can see so many research being going on in this zebrafish, right from the embryonic stage to the adult stage. It has wider applications like all other animal models. We cannot neglect this as a other model, but it's a very good model. I hope you would look for zebrafish studies, and you can also look for zebrafish on the internet as well. And if you have any other kinds of doubts, you can post a comment here. Otherwise, you can also send a message to my email ID there provided. So thank you very much for watching this.